Hello everyone, in this video I will demonstrate how to count cells using a Bio-Rad analyzer. For that we will need to mix equal amounts of our cells with trypan blue, which is a staining to stain cells. We got our cells from trypsinization or from scraping of the cells, which is in a 15 milliliter tube. And now we're going to grab ourselves Eppendorf tube, where we will mix our cells with the trypan blue. So there's different ways on how to get an Eppendorf tube from the box. And if you have tweezers, you can put the tweezers above the flame for a few seconds. Wait until it cools down. And then grab an Eppendorf from the box. Of course, think about the oops rules. Once you open the box, don't go with your hands above the tubes. Another way, if you don't have tweezers, is lightly shake the tubes until you get one Eppendorf tube on the lid of the box. Then you can grab the tube, close it, and then close the box. And now we have our sterile Eppendorf tubes. And now, as I mentioned, we need to mix equal parts of our cells with the trypan blue. I will be mixing 10 microliters of trypan blue with 10 microliters of our cells. So I get my trypan blue, 10 microliters. And now I get my cells. Remember to first mix them before pipetting. Otherwise all your cells will be at the bottom, and if you pipette from the top, the reading will say that you have no cells, but you do have cells, they're just all at the bottom. So make sure to mix before pipetting. So I also pipetted 10 microliters of my cells, and now I will mix it with the trypan blue. Pipe it up and down a few times. Now we have our mix of cells and trypan blue. So now I will get 10 microliters of my trypan blue cell mix. And I will add it to this counting slide. Hope it can be appreciated, but you have sort of these half moons where you can put your pipette in and then just pipette it out, and through capillary action your sample will go into this rectangle. And now you can bring this light to the Bio-Rad analyzer. So this small machine is the Bio-Rad analyzer. We have our counting slide here, and the part with the cells that we want to count, we introduce it. And then we just wait a little bit until it's done counting the cells. So it is now done counting, and we can see on the small screen different values. We're interested on the live cells, which is a percentage of live cells that it's counting, which is an indication on how happy your cells are, but also how well you might have scraped or trypsinized your cells. So write that number down. And we're also interested in the live count. So this is the amount of cells that are alive in your sample per milliliter. In this case we have 4.22 million cells per milliliter. And this is also the number that you need to write down, because you will need to calculate dilutions with this number. And that's how to count cells.